All right. Hello, everybody. Can you hear me? Good. Okay. Thank you all for coming to the DeFord seminar this lovely weather Friday. It's my pleasure this week to introduce Marjorie Cantine to you. She did her undergrad degree at Wellesley College, her PhD at MIT alongside me. She graduated a bit before I did. And then she was a postdoc with a prestigious Marie Curie Fellowship at Goethe University in Frankfurt before starting her current position as an assistant professor at the Earth and Space Sciences Department at the University of Washington. Marjorie does some really interesting work focusing on the geochemical and physical records of time and environment preserved in rocks. And today she's going to talk to us about that work. So I'll not talk to you any longer and I'll pass you straight over to Marjorie.
Thank you, Craig. And thanks to all of you for being with me today. I'm really excited to get to share a little bit about my work with you. It's been a wonderful visit so far. And yeah, just thrilling to spend time with so many people who think about and care about the records of time and environment that get preserved in sedimentary rocks like the ones I'm showing on this slide. So this is one of the field sites that I'll be talking about today. This is Mistaken Point up in Newfoundland, which is a UNESCO World Heritage Site where some of Earth's first animal fossils are preserved on bedding plane surfaces. Sedimentary rocks like the ones I'm showing here preserve precious geochemical, physical, and fossil records of what Earth's surface environments were like in the past. And they're sometimes our only record of what those environments were like. But to make full use of those records, we need to be able to place them in time. So the work that I share with you today is going to be about doing that, why doing that is difficult, and why it's important to do it well. This work has benefited from the involvement of so many different people at different institutions, as well as community partners up in Newfoundland. And I particularly want to acknowledge the contributions that I'm showcasing today here, most of which come from my PhD student, Hannah Cothran, who's really been spearheading this work. When I look out around this room and I think about what all of us work on and I think about how time is a master variable across the earth sciences. Kind of no matter what you work on, you're thinking about time in some way, whether you're thinking about earthquake recurrence intervals, geohazards along the western coast of the United States. If you're thinking about the interaction of Earth's chemical reservoirs captured here and looking at the diffusion of various chemical species through different mineral and fluid phases. If you're thinking about large events in the history of life, like mass extinctions or origination and evolution, if you're thinking about the origin and evolution of planetary bodies, whether this Earth or other ones elsewhere, or whether you're thinking about teleconnections, the ways that our climate changes on human or slightly longer than human timescales, how one place can get warmer and wetter while another gets cooler and drier. Time is required for putting the inherently regional records preserved in sediments into a true broad global context so that we can understand spatial variability in Earth's complex and co-evolving systems. Time informs all of these different questions, taking us from the atmosphere down into the Earth interior. And we have to think about time at a variety of timescales, right? There are events represented on this slide that range from a few seconds to events that took place billions of years ago. And so we think about this really broad range of timescales in the earth sciences. And time unites all of these different areas of focus because time is uniquely capable of helping us understand things like the frequency and the repetition of rare events, which is critical for things like geohazard predictions, but also critical for understanding things like mass extinctions, impactors, rare events in Earth history. Time is critical if we want to calculate fluxes, things like the exchange of redox potential or energy or chemical species between different reservoirs on our planet. Time and the ability to place ordinated events on a timeline is also critical for understanding and decoding sequences of events, understanding what came first and what came next, disentangling cause and effect. And that's required if we want to understand process and result. This is also, the ordination of time is also critical for building truly integrated global records that allow us to understand how the planet varies both spatially and temporally. And I think that this recognition of the importance of time, as well as our recognition as a community that our planet has a really deep history that extends some 4.5 billion years into the past, sets our field apart from others. Perhaps cosmologists come close to this, but I think that this is really an area where Earth scientists have something unique to contribute, both to our the sort of broader scientific community and to public discourse. As Earth scientists, we're almost uniquely aware of just the degree to which human beings occupy this sort of very thin bleeding edge of this really deep spiral of Earth time, deep time that extends some 4.5 billion years into the past. This awareness of deep time is something that we find it really easy to talk about with each other. But if you've, I don't know, I had a great meeting with Bara today where she said the Cretaceous wasn't that long ago, right? That's something that only an Earth scientist would say. And I love that about our field. I think it's a really important and special perspective that we bring. Much of our own geological intuition and the intuition that we try to instill in our students comes down to recognizing time in space. It comes down to recognizing relative time relationships, what's older and what's younger, from the way that geological materials are organized in space. So folks who teach here in the Amazing Field program that exists here are always developing this understanding in their students. One way this is reflected that's particularly close to my heart as a sedimentologist is stratigraphic superposition. The recognition that younger layers of sediment accrete on top of older layers of sediment. And so there's a relative time relationship recorded in every strat column. Structural field geologists like Craig will recognize cross-cutting relationships in the field and use them to ordinate events in time and think about processes of deformation and change encoded into the surface expression of geological materials. And a lot of our studies of Earth processes focus on processes that extend for much, much longer than any individual Earth scientist's career. So being able to recognize how those processes vary in space and use clever substitutions that swap in space for time allow Earth scientists to study those really lengthy processes. So for example, you can imagine geochemists sampling along the track of a hotspot island chain, swapping in time for space there. Or you might imagine ocean-going geologists sampling at various distances from mid-ocean ridges to sample lithosphere of different ages to gain a better understanding of this process, which lasts too long for anybody to sample it in real time. This recognition of space for time swaps turns the surface of our planet into a natural laboratory where clever and exciting work can be done. In addition to and beyond these relative time relationships, we've worked really hard as a community of Earth scientists to develop ways to measure absolute time. So not just what's older and what's younger, but quantitative time, numerical time. So one way that finds expression is in the geological timescale. I think it's tempting to think that this was handed down on stone tablets at Mount Sinai, but of course it wasn't, right? The geologic timescale is a labor of love put together by scores of scientists working together over the years to articulate a truly global temporal framework into which regional and local observations can be tied together into a unified story of our planet. That geologic timescale is often anchored by radiometric dates, so dates on geological materials that take advantage of the decay of radioactive elements into their daughter products, and the calculation of absolute dates from that. Those techniques exist alongside tools like astrochronology or magnetostratigraphy, which are also ways of thinking about and telling time. And an immense amount of work has gone into making all of these different ways of telling time talk to each other in a unified and interoperable way, so that the way that you tell time in one system agrees with the way you might tell time in another system. Where it becomes really challenging to measure absolute time, there are still people making clever use of things like constant flux proxies to try and back out the ways that sedimentary environments change through time and to calculate rates that are sort of independent of the sedimentary noise and intermittency that can affect sedimentary records. And the title of this recent consensus studies report from the National Academies, I think, reflects this recognition that time is really important. Across all timescales, no matter what part of the Earth system you work in, we are all thinking about critical issues that involve the Earth in time. So welcome everybody to the middle of the Earth in Time decade, 2020 to 2030. This broad focus on time across our community recognizes that Earth history and Earth processes have to be interpreted from incomplete records using imperfect tools. We wouldn't have spent so much time developing this understanding if this was easy and obvious. But we've spent so much time on it because it's hard and because it's important. Today I'm going to tell you about my work at the frontier of some of these questions and controversies to better correlate and better understand Earth's first animal ecosystems. So this is a photograph of one of my field sites at Mistaken Point that I mentioned earlier. And the beautiful and enigmatic fossils that we see preserved on this bedding plane we'll return to later. These are members of a group of organisms called the Avalon fauna, which represents Earth's first diverse animal ecosystems, preserved in Ediacaran or latest Precambrian sediments. But before I get there, I need to tell you a little bit about the tool that I'll use to ordinate the Avalon fauna in time. High precision dating of a mineral called zircon is often referred to as the gold standard in absolute dating of Earth history. This is an image of what zircons look like. They're often found in volcanic ashes, tuffs, bentonites, and once you remove them from the surrounding matrix, this is what they look like. They're sort of these sand-sized prismatic grains that, as they form in a magma chamber, incorporate substantial amounts of uranium and very little lead. That uranium decays into lead at a well-understood rate, and so a measurement of the uranium and lead ratio in a single zircon can give us information about the timing of the crystallization of that mineral. This work is incredibly sensitive, analytically, difficult, and long. It's really sensitive to contamination by lead, which is ubiquitous in the modern environment. So all of this work happens under clean lab conditions in highly specialized laboratories. It's incredibly finicky work. It involves the manipulation of these tiny grains of sand effectively, their chemical abrasion, their dissolution, and then their analysis. This is painstaking work. My student Hannah has put an immense amount of time into it, and I'm grateful for that. Within each individual zircon, we're looking at the decay of two flavors or two isotopes of uranium, 238, which decays to 206, and 235, which decays to 207 lead. So these two isotopes of uranium are both incorporated into zircons. They decay at their own rates, and they provide a natural cross-check upon each other. So we can understand if the zircon is recording a reliable age by comparing the 238-206 age to the 235-207 age. If they agree with each other, the zircon will plot along the orange curve I'm showing here. Here we have our two uranium-lead decay chains. This curve is called Concordia, and a zircon that behaves well and yields a reliable date will plot somewhere along this curve. The numbers that you see plotted along this curve are ages in millions of years. And here's the part of Concordia that we'll be getting up close and personal with today, in the age range of about, say, 565 million years ago to 585. This technique, like I said, really makes up the backbone of the geologic timescale. It is widely used, and to demonstrate that to you, I have assembled this pie chart of the nearly 400 dates that go into constructing the geologic timescale older than 1 million years. Everything you see in blue was measured using the uranium decay system. The vast majority of that on zircon, and the vast majority of that with this technique called chemical abrasion, isotope dilution, thermal ionization mass spectrometry, or CAID-TIMS. So this is that very painstaking technique that takes place under clean lab conditions. And the reason that it makes up such an enormous chunk of this pie chart is that the precision possible with this technique is truly remarkable. And you'll see that reflected in some of the data that I'll show you later. We have invested a tremendous amount as an earth science community in being able to date this mineral, zircon, at increasingly high precision. And to illustrate that to you, I'm going to show you how our understanding of the age of a particular unit has changed through time as a result of increasing analytical precision, increased investment in this technique. So I'll show you how our answer to this question, how old is the Fish Canyon Tuff, has changed through time. The Fish Canyon Tuff is a volcanic unit that is an important source of multiple mineral standards across a range of dating techniques. The first study that I'll show you here answered this question using fission track dating, which is not directly a radiometric dating technique. And it answered this question with a precision of a little bit worse than a million years. This is all 95% confidence intervals. So the length of this bar represents the precision of the estimate. As techniques improved, you see that we start with dating multi-grain zircon analyses. We eventually get down to dating single zircon grains. We continue to improve precision. The bars get narrower and narrower until finally we are able to date individual zircon grains at incredibly high precision. And I'll just highlight here the difference, 3 orders of magnitude in the precision between the first study and the last study shown on this slide over a period of about 30 years. I wish that I could get 1,000 times better at anything in 30 years. That's pretty incredible. But as we have improved the precision here, we have encountered other issues. And that's reflected with this statistical metric called MSWD, which is a statistical, or it's a parameter that allows us to think about how many different populations we might be sampling with our data. So when the data is relatively imprecise, it could all be explained with merely analytical scatter associated with a single age population. But as our precision improves, that MSWD metric creeps up, indicating to us that we are in fact sampling multiple separate populations. We are now able to resolve the ages of individual magma chamber processes and events. And this really complicates the question, how old is the Fish Canyon Tuff? It's no longer one age. There are multiple processes here which can be measured and dated. And this creates problems for chronostratigraphers, for myself, who want to date a volcanic ash and use the age of that ash to constrain the ages of sediments directly above and below it. If we sample this ash and we get this really complicated, rich record that is telling us a lot about magma chamber processes, and we don't recognize that complexity, we set ourselves up for a lot of confusion and potentially failure in interpreting the depositional ages of sediments associated with these volcanic ashes. And this complexity reflects the fact that even though chronostratigraphers are really interested in dating zircons interbedded in sedimentary rocks, zircons are fundamentally igneous minerals subject to igneous processes. And they can be affected by a wide range of processes that occur before the volcano ever erupts, spews ash, which then deposits and is incorporated into a stratigraphic section. But zircon isn't just an igneous mineral. It's also affected by sedimentary processes of erosion, reworking, redeposition, abrasion that can all occur after. And if we fail to honor this geological complexity that's present in the materials that we're dating, we may really misinterpret the records of time that we are trying to use zircon to try and tell. So as a community, this is where I think we're at. How do we detect and notice this geological complexity in our samples? Are there new screening techniques that we can use? Are there analytical tweaks to our analytical methods that we can make to better detect and characterize this complexity? And once we've noticed that complexity, what do we do next? How do we deal with it? How do we integrate it into the story and the timeline that we're setting up? And given that given that our analytical precision now in some cases may outstrip our geological understanding of the system that we're working in, what questions, what locations and settings are most tractable and most ambitious given the precision and the accuracy of the techniques that we can use? So now I'm going to tell you a little bit about the time and place that I've elected to tackle these questions. And to do that, I need to tell you about the avalon fauna. These are members of Earth's oldest ecosystems that include animal-grade organisms, so macroscopic, multicellular animals with tissues. These are pictures of some of the avalon fauna from Mistaken Point and other locations in Newfoundland, which is the oldest place on Earth they've been described. In all of these images, we're looking down at a bedding plane, and these strange and enigmatic and quite beautiful impressions that you see are the avalon fauna. They have these often semi-fractal body plans. They lived anchored in place on the deep sea floor, and as far as we know, they have no living relatives. They were soft-bodied with a texture maybe of something like a jellyfish. And so their preservation in such detail is really quite remarkable, quite special. And yeah, this is what we think animal life looked like when it first appeared on our planet. The avalon fauna is known from Newfoundland as well as from northwest Canada and from Charnwood Forest in the UK. This is one artist's impression of what the avalon fauna might have looked like in life. Here's another artist's impression. These reconstructions highlight the fact that the avalon fauna largely predate locomotion, largely predate predation. So these are sessile, benthic-dwelling organisms that live anchored in one place on the seafloor. They're not moving around. And the model for their preservation in this really spectacular way of these raised impressions on beds is due to their unique volcanosedimentary depositional setting. So as I said before, these organisms entirely lacked hard parts. There's something with the texture of a jellyfish or a sponge maybe. And so the model for their preservation is that you have an avalon, a member of the avalon assemblage, living anchored in place. It's buried by a turbidite or perhaps by a volcanic ash flow. The organism decays and sediment from the turbidite bed below fills in that mold, creating a cast that we see in relief here. So this mode of preservation gives us these sort of raised impressions spread across bedding plains that once represented the deep seafloor. They lived at depths of hundreds to thousands of meters, often capped by volcanic ashes, which fortunately for us contain datable zircon. The other thing about this mode of preservation that I think is really exciting and really scientifically interesting is that these organisms are preserved where they lived, and they're preserved with, we think, near census fidelity. So it's like a snapshot of a community in situ. And that means that these bedding plains allow analysis with all sorts of really interesting paleoecological modeling that generally can't be applied to the fossil record. So my collaborator, Emily Mitchell, has been at the forefront of this work. She goes out and she maps these bedding plains with a laser, capturing the fine details of every organism upon them. And from their spatial relationships, she's able to recognize things like specific interactions between different taxa. She is able to identify certain poorly preserved forms as likely taphomorphs of other organisms or likely the degraded remains of other specific taxa. We've also gained insight from the relative sizes and distributions of these organisms into the reproductive modes of animals that are more than 500 million years old, which I think is a phenomenal insight into the life histories of long extinct organisms. We're able to understand ecological succession after the disturbances of these turbidity currents or ash flows. And we're able to look at things like tiering and resource competition between these different taxa, how they interacted with each other and with their environment. And overall, when we compare the Ediacaran seafloor, the seafloor of the Avalon fauna, we find that there's a lot less competition there than in modern benthic deep marine environments. So this is sort of a life as we don't know it snapshot of a world that looks really different from our own. I think these insights are phenomenal. They're really motivating to me. They're really exciting. The sort of insight that we get into the life histories of early animals from these observations are just astonishing. And yet there's still fundamental questions about why these organisms show up when they do, and if there's some connection with environmental triggers for their appearance. Remember that for most of Earth history, that long spiral of time we looked at early in this talk, about 90% of Earth history, life on Earth is microscopic and microbial. Why life gets big and macroscopic and multicellular when it does is an open and interesting and an important question in both the Earth and the life sciences. Some have hypothesized that there might be some sort of environmental trigger that permits the evolution of these larger and more complex animal species. There's also a lot of other change happening in Earth's environments alongside the appearance of the Avalon fauna. So there's evidence for glaciation in Newfoundland at around 580 MA. I'm showing you here the Gaskir's diamictite, which is a deposit left behind by glaciers. Some have hypothesized an environmental connection between this cold period and the appearance of the Avalon fauna. There's also a really strange geochemical signal preserved in sediments of roughly the same age. Earth's largest negative carbon isotope excursion, which I won't get into too much today, but this very strange geochemical event that has kind of puzzled geochemists and Earth historians, which appears to be roughly the same age as the appearance of these animals. And that geochemical signal is sometimes interpreted as evidence for oxygenation of the ocean at around the same time. So when people think about an environmental trigger for the appearance of these complex animal forms, they often suggest that the ambient oxygenation was a fundamental limiting control on the appearance of complex animal life. The reason that it didn't show up for so long was because animals use oxygen to do aerobic respiration, and they were fundamentally constrained by ambient oxygen levels. That's a really interesting hypothesis that has come under renewed scrutiny as people develop both better paleo-redox proxy records and more sophisticated ways of analyzing those paleo-redox proxy records. But our ability to sort of ask and answer the question of how and if these environmental changes might be related to the appearance of early animals has been frustrated in part because we lack robust age control on these early animals. And to illustrate that for you, I'm going to talk about how we've been living with two mutually exclusive ages for these first animals on our planet. And to do that, I have to introduce you to something called the pizza disc bed. So the pizza disc bed is a famous fossil surface at Mistaken Point, and it's called the pizza disc bed because it contains this type of fossil called an Ives hediamorph, which people sometimes think looks like a pizza. Elsewhere on this bed, we also find things like Sectardus, which is sometimes interpreted as an early sponge. And elsewhere, we also find these beautiful little frondlets, which are interpreted as juvenile forms of some of the Avalon fauna that we've already met. So this is a classic fossil surface, and it's capped by a 35 centimeter thick volcanic ash, which has obviously been an exciting target for folks trying to understand the timing of the appearance of these organisms. So the first age published on these data, or published on this ash using modern chemical abrasion ID TIMS techniques, was published in 2016, and it came out with this age of about 571. Another study dating the same material, using the same technique, came out with an age of 574. And you might be tempted to think, well, what's 3 million years between friends in the Precambrian? And I would say 3 million years matters a lot when you're thinking about cause and effect relationships, when you're thinking about questions of evolution, speciation, and most of all it matters when you're using a technique that underlines more than 3/4 of the geologic timescale, which says it should agree within a few Milankovitch cycles of each other, but you're not able to resolve differences of several million years. So the problems here start with different disagreeing ages on this bed, but they don't end there. I'm going to show you the different age models implied by these two studies for these strata. So on the y-axis here, we're looking at the stratigraphy. Stratigraphic height is here. Here is the age model implied by that first paper I showed you. So this is new age modeling that we've done using modified Bcron, which is a Bayesian age depth modeling software package that integrates our assumption of stratigraphic superposition, that is, we should be getting younger as we move up section, and couples it with measured ages that are defined by the user. This allows us to interpolate the ages in a quantitative, reproducible way for every bed in this stratigraphy, even beds whose age we don't measure directly. So this is one age model. This is the other age model implied by the stratigraphy. And then this figure captures the differences between these two models. So if these models agreed with each other perfectly, they would align along this vertical line. They don't. They disagree substantially. They predict very different ages for key fossil surfaces throughout the strata, including the very first fossil animals, which occur several meters below that famous pizza disc bed. And all of this with a technique that says it has an error encompassed by this error bar here. So 3 million year difference using a technique that claims far sub million year precision. So what's going on here? Is this geological scatter? Are these those magma chamber processes causing complex age spectra to be recovered in our data? Is there evidence for sedimentary reworking and complication of our ashes that's been previously unacknowledged? Are there analytical issues here that are affecting one of these dates? Are there sampling issues? Are we even certain that these studies are sampling the same bed? All of these questions were on my mind. And so I'm excited to give you an answer to some of these questions, thanks to Hannah's work applying new screening and modeling techniques to bring resolution to this and other questions. So the sort of state of the art of CAID TIMS for a long time, you see these very low N numbers here. So this is an age measured on 5 individual zircon grains. This is an age measured on 9, and then a subset of the youngest 4 chosen. This low number of dated grains reflects the incredible time commitment and expense that this high precision technique requires. New screening techniques that have been developed by our collaborator, Mark Schmitz, and his team at Boise State takes a totally different approach. So at that lab, they analyze a suite of zircon grains. So for example, in our study of the pizza disc bed, we analyzed the ages of about 100 of these grains from this bed at relatively low precision and relatively rapidly using laser ablation, which is a lower precision technique. At the same time, we also captured geochemical data that helps us think about the geochemical populations that we see. So once we have that screening data set, we're able to use it to identify a subpopulation of the youngest geochemically similar grains and advance those through to the more time-intensive technique. We also image every grain with CL and BSE, which helps us examine the potential for things like reworking and abrasion, which we would expect to modify the shape of these grains. And we only select for further analysis grains that we think truly represent ashfall zircon that has not been modified by subsequent sedimentary processes. And after all of that is said and done, this is the age that my team recovers on this famous pizza disc bed. So we have an age here of about 571. We overlap within uncertainty with this first age published here. We think everybody sampled the same bed. This is a really classic fossil surface. It's capped by a really distinctive ash. It's the only one that matches its description in the area. We think that everybody is, we're not convinced that there's an analytical issue here, but what we think is going on are real geological complexity in the materials that we're analyzing. Our collaborators see spread of several million years in volcanic ashes of broadly similar age, which they attribute to these magma chamber processes. The crystallization age of a zircon is something different from the time at which it's erupted and deposited. And we interpret this older age as reflecting an older population of grains that is present in these rocks. Furthermore, our confidence in this younger date is bolstered by an additional date we measured 125 meters below this pizza disc bed. So in accordance with stratigraphic superposition, we should expect this lower age to be older, and that's exactly what we find. Critically, this ash is also older than than that age we now interpret as erroneously old on the pizza disc bed. And so we have about 1 million years of time difference between these two ashes. In this ash, 125 meters below. So we're obeying stratigraphic superposition, getting younger as we get older. And what I'd critically like to point out here is that the very first fossils at Mistaken Point occur about 3 meters above this ash. So this pizza disc age has often been propagated through the literature as an age constraint on Earth's oldest animals. It's not. This age is our first high precision constraint very close stratigraphically to the lower end of these Avalon fauna communities. I can't believe that we lived without it for so long. So our reevaluation of existing age models, the data that's out there, and our own new dates lead us to this age model for the strata at Mistaken Point. So this is an age model that I'm now really motivated to go out and continue to test. The results that we got surprised me, and I'm excited to continue to test and refine this age model by dating new ashes through these strata. The really amazing thing about these Bayesian age depth models, what they allow you to leverage, is the fact that the date of every ash constrains the date of every other ash in the strata because of stratigraphic superposition. So this is a predicted model that we can continue to test and refine. And the true power of this age model is what it allows us to understand about other locations. Now that we have this better resolved temporal framework for Mistaken Point, we can now critically examine fossil occurrences at other locations. So all of the work that I've told you about today so far has been down here at the southeast tip of the Avalon Peninsula at Mistaken Point. The similar fossils, similar age, are also exposed up here at Catalina Dome. So this is another famous fossil site. It's on the Bonavista Peninsula, and you can see that there's about 200 kilometers of distance between them. Despite this distance, despite the fact that there's no exposure between these sites except at the coastline, despite the fact that Newfoundland is structurally very complex, these two locations are correlated with each other on the strength, canonically, of lithostratigraphic correlations in what are quite ambiguous and monotonous turbidite sequences. So that canonical correlation between these two locations is shown here. The formation boundaries were sort of applied to Catalina Dome, and they are assumed to be contemporaneous between the two locations. If you take this canonical correlation at face value, this is what it implies about the different fossil distributions between these two sites. So the Mistaken Point fauna distributions are shown in black, the Catalina Dome distributions are shown in gray. And if you take this at face value, it implies the fossils at Catalina Dome are systematically offset younger than fossils of the same group at Mistaken Point, implying an offset in time and space that has been used to inform our understanding of how these communities change through time. There was some pilot data in the literature that made us question this canonical correlation. And I'm pleased to share some early results that support the idea that this canonical correlation is worth questioning. Here we're looking at the Melrose surface. This is the stratigraphically highest, so the youngest diverse fossil surface in Newfoundland. You can see it's totally full of fractifususes across this bed here. There's another thick volcanic ash that sits on top of this bed, which yields a date of 565 MA. So this date, in fact, far from taking this surface younger than anything at Mistaken Point, actually sticks us right into the Mistaken Point stratigraphy. That, combined with a developing age model, these ashes are quite complex, we're still making sense of them, allows us to correlate this fossil surface back into Mistaken Point. You can see that we land within the fossil-bearing strata at Mistaken Point. So far from this fossil surface being substantially younger, with no coeval equivalent exposed at Mistaken Point, we are now able to compare fossil communities in space and time in a more meaningful way. So these results move us away from this canonical correlation based on nothing more than lithostratigraphic boundaries into this alternative hypothesis, which brings the temporal distributions of these groups into better alignment. Instead of having a systematic temporal spatial offset between these two sites, they come into focus together. And I would argue that this is critical for understanding how and if these communities vary in space and time. It's only with this chronostratigraphic framework that we can make truly meaningful comparisons of coeval communities. So these results give us new clarity on the rise of animal life on our planet. These results now constrain the first appearances of the Avalon fauna to about 572. This is the age of the ash, just 3 meters below some of the first fossils. And this makes their first appearance roughly coeval with the onset of the Shuram excursion, which is that very strange geochemical signal that I spoke about earlier. So my work on the Shuram excursion in Oman, which is the classic sort of type section for the Shuram excursion, used rhenium-osmium data to constrain the onset and the termination of the excursion. This is the age of the onset. You can see that we are within uncertainty of the technique, roughly coeval for the first appearance of animal fossils with the onset of this very strange geochemical signal. So does this support the hypothesis that the Shuram excursion represents an increase in ambient oxygenation, which was potentially a key threshold that needed to be surpassed for animal evolution to occur? It's a hypothesis, and it's one that my team is excited to continue testing. I think Newfoundland is the perfect place to test that hypothesis. We have this almost world-unique location that has both abundant fossils of the right age and type and abundant ashes. To my knowledge, there is no succession of the right age that has such abundant ashes and fossils that allow us to build out the robust temporal framework needed to test this hypothesis. What we currently lack in Newfoundland is a really robust redox proxy work using some of the latest paleo redox techniques. And that is work that I'm excited to continue doing with my collaborator, Chad Ostrander at the University of Utah. When all is said and done, I think what we'll have in Newfoundland is exactly what we need to test this hypothesis that oxygenation was intimately tied to the rise of animals on our planet, and the hypothesis that the Shuram excursion represents some sort of oxygenation event. To test that hypothesis, we need a robust chronostratigraphic framework, we need fossil appearances, and we need a robust redox record. We've made substantial progress on 2 of those 3 things, and I will be excited to share my results in the years to come. Another important aspect of this work is the ability to correlate coeval fossil surfaces precisely, rather than relying on canonical lithostratigraphic correlations to tell stories about changes in animal diversity through space and time that may not actually be spatially or temporally accurate. So to wrap it up and bring it home for you today, I just want to leave you with a few messages. Time is a master variable in the earth sciences. Being able to recognize it and measure it accurately and precisely is fundamental to the work that we do. Whenever you gain new levels of precision with any technique, whether that's spatial resolution or analytical resolution, you are forced to consider geological complexity in new ways. For geochronologists, what that means is that these increasing levels of analytical precision have forced us to contend with complicated geological processes that before now we couldn't resolve, and so we didn't have to consider in our application of those techniques. We're now at a threshold where we're beginning to consider the rich complexity that those processes introduce into our records. And I also want to celebrate and highlight Hannah's work dating the rise of animals on our planet with more precision and more resolution than ever. We're so excited to continue this work to better understand the rise of complex animal life on our planet, what it means for all of us sitting in this room as complex life living on this planet, understand the fundamental constraints that shaped animal life at its beginning, and understanding what, if any, environmental trigger was associated with its rise. Thank you so much for your time and attention. I'm looking forward to your questions.
All right, thanks, Marjorie. We're going to continue the new tradition here of having students ask questions first. So if you're a student in here and you have a question, now's the time. Yeah.
So in the beginning, you talked about screening those 100 grains for LIS and PMS before choosing them for ashes. Did you guys bulk mount? Like, did you take that as a bulk stuff, or did you handpick that? And then did you handpick that based on like anything you did? What kind of?
They were handpicked. Yeah, they were all handpicked, and they went through chemical abrasion, and then they were mounted. And they were picked based in. Yeah, morphology plays a role in how they're selected. We're looking for grains that look, look volcanic, look primary, look not reworked, and that is captured, I think, not really at the mounting stage, but really at the imaging stage. So after mounting, when they're imaged and characterized.
I honestly don't know much about this kind of stuff, but you mentioned this geologic complexity. Can you ask questions about geologic complexity, like how does the volcanic evolution, because you're able to meet like these different stages, change in an area? So like, I have all these volcanic layers and all these different zircons that look at the different phases of volcanic activity, and how does that change in an area?
Mm-hmm. Yeah, absolutely. That is certainly a question that could be addressed with the data set and the samples that we're collecting. Our focus at the moment has really been on the chronostratigraphy, but we've wondered about the possibility of doing some more robust or some additional high precision dating of some of the geochemical oddballs that we see in our samples to see if that partially explains some of the unusual older dates recovered by that one study. We wonder if they were really just sampling sort of a distinct and weird group of grains.
I'm in the same vein as Jack's question. When you're going and looking at all these grains, you said you're going to like the ones that aren't important, but you're
Mhm.
the ones that are really igneous.
Yeah.
Are you able to like distinguish between like older population and the igneous population, or are you mostly just looking?
Yeah, so with the laser, at the scale of the laser, like at the precision of the laser in this particular system, the grains all fall within a single population. We do get some like the older flyers. We don't analyze further. So within if you look at Hannah's data, you see that some. So if you look at the Melrose ash here, you can see that resolvable with chemical abrasion, ID-TIMs, we do see some of those older grains get included, but with the precision of laser ablation, these would all be the same age.
So if that's all the student questions, you can open the floor to everybody else if you have a question. Now's your chance. Yeah.
I'm just curious about this claim of near census fidelity in the fossil record. How do you confirm that?
Yeah, that's a great question. So I think that the most robust way, or I think evidence that suggests that is true, is that we have preservation of various taphomorphs in various states of degradation. So these Ives hediomorphs, which represent the decayed remains of some of these fossils, are preserved, and we see a gradation in how well they are preserved, like from very early states of decomposition all the way through more extreme states of decomposition. So I think that's interpreted as evidence that we are capturing a seafloor community sort of as it really was. These organisms are also, they're preserved in such a way where if this cast and mold filling model, how do I want to say this? I think so. They all have this sort of similar texture, similar sort of body plan, living flat or erect on the seafloor. And I think that if you want to argue that there's like a systematic taphonomic bias, I think it's hard to reconcile exactly what that bias would be with the density and the preservation of fossils in this specific taphonomic mode. I agree with you that near census is difficult to quantify, but we do see, I think, really remarkable observations like these big fractifususes surrounded by little baby fractifususes which seem to bud off from them. We see things like these rangeomorph fronds with these baby rangeomorph fronds behind them and what the paleontologists call conga lines, which are interpreted as sort of being deposited in the lee of the parent frond. And so we observe these ecological relationships that, certainly are interpreted as being meaningful.
Who's going first? Do you think, since you find these only on bedding surfaces, that these are actually organisms that are brought down with the currents and pioneer these places? So they're coming from shallower water?
That's an interesting question. I don't think so. I think that these are genuinely. So I guess there are sort of two things I hear at that question. One is what does the shallow water community look like at this time, and are these being transported in, or are they living in deep water environments? So in northwest Canada, that's where these organisms are also found. You have facies that would tend to preserve these organisms in shallow water and in deep water, but these organisms appear in the deep water first and they are absent in the shallow water equivalents. And so that's the case in northwest Canada, and then in Charnwood Forest and in Newfoundland,